Hello, welcome to another screencast where this time I'll be discussing the microanatomy of skeletal muscle. This is the second part of this, and this is going to have to deal with looking at and identifying and describing here the structure of a skeletal muscle fiber. So now we're at the cellular level, so we're looking at the muscle fiber, and we'll talk here about the sarcolemma and these T-tubules. Also the intracellular structures, the organelles, if you will, of the muscle fiber, so sarcoplasmic reticulum, these things called myofibrils, as well as then sarcomeres. Then we'll spend a lot of time here looking at the structure of the sarcomere, and then all of this alphabet soup of things here, so the Z-line, or sometimes called Z-disc, the A-band, the I-band, the H-zone, the M-line. And then finally, detailed super microanatomy here of the myofilaments, the proteins actin and myosin. So both of these are the proteins of muscle contraction. So let's get started here. Pardon me. So it's seen in the previous screencast about skeletal muscle that it is striated under the light microscope, and so these are the muscle fibers. Again, here they are long cylindrical cells, and we see very clearly here the stripiness here, the striations of those cells. And then here it is in cartoon view down here. So what we're going to do is I want to look at what this cell looks like here at the inside, okay? And so we're going to do this, just a quick review here. Again, here's your bone, here's your tendon. We talked in a previous screencast about the muscle structure itself and how the muscle is actually groups of these fascicles, but then each fascicle or fasciculus that I pull out there is actually groups of skeletal muscle fibers. So here we've seen that we've pulled out one muscle fiber here, and we have artery capillaries that are on the surface of that cell, and there are several nuclei that are peripheral, right? And this is covered then by the cell membrane. So this is the plasma membrane, and that is our first stop here, is the sarcolemma. The sarcolemma is the fancy term for the plasma membrane or the cell membrane, and so this is the cell membrane of the skeletal muscle fiber here. And I want to point out a feature here of the sarcolemma is that the sarcolemma is continuous with these, this membrane is kind of folded down inside of the cell here, and those invaginations, those infoldings, are the transverse tubules or the T-tubules. And they're going to be important when we look at the functioning of this and the physiology of the skeletal muscle. So we'll come back to that and deal with that anatomy a little bit later as well. So here you see that we've taken a section away from the skeletal muscle cell, the muscle fiber. The sarcolemma has been removed, exposing the inside of the cell. And this yellow structure here is the sarcoplasmic reticulum. The sarcoplasmic reticulum, which is really the endoplasmic reticulum of the skeletal muscle cell. And again, so sarco is flesh here, having to do with muscle. So the sarcoplasmic reticulum, again, is going to be important in the physiology of the muscle, so just hold on to that. We see that inside of the cell, then, the nuclei have been pushed to the periphery. Also then, there are going to be loads of mitochondria inside of these cells as well, because the mitochondria are places of ATP production, and when the skeletal muscle cell contracts, that ATP is going to be converted to ADP and an inorganic phosphate here, thus releasing energy out of this. So this is energy comes out comes out. Energy is provided for the contraction there. The rest of the cell cytoplasm here is made up of these highly repeating units of proteins, and they are themselves grouped together in small little bundles called myofibrils. So the myofibrils are these circular areas here that the T-tubules are sort of surrounding. And so here we see 1, 2, 3, 4, 5 myofibrils. We've pulled that myofibril out. And what I want to point out here is that this is where the level at which you see the stripes, right, the striations. And so those striations are due to the repeating patterns of the sarcomeres here. And so a sarcomere is going from this to this, and then I have another sarcomere here and another sarcomere here, so this would be a sarcomere, and that would be a sarcomere, and this would be a sarcomere here, right? So sarco, flesh, mere is a unit, mer is just a unit, so this is the flesh unit, so you have this from this border to this border is the sarcomere. So now if we zoom in on that, what we notice is that the sarcomeres are made up of these repeating patterns of two proteins that we call myofilaments. Right? So a myofilament actin is a thinner one, and then the myofilament myosin is a thicker one. So actin is thin, and myosin is the thicker of the two. And again, these are both proteins that make up the skeletal muscle fiber. So when you are eating muscle, that is beef or chicken or fish or any of the muscle of any animal, it's high in protein, and these are two of the main proteins that you're eating there, the actin and the myosin. So now we're going to zoom in on one of these sarcomeres here, and we see that the sarcomeres are bordered by this Z disc, right? And then a Z disc over here. This is sometimes also just referred to as the Z line. I think you might be able to see why they call this the Z line, because it zigzags here. And so anchored to this protein, and so this Z line, this Z disc, is made up of a separate protein, an actin and myosin. But this protein here anchors the actin molecules, which are seen in purple. And then there are these titin proteins here, and these proteins are also attached to the Z line, and then they are attached to the thicker filament here, which is in green, and that is the myosin molecule, the myosin and myofilament molecule. At the center of that myosin molecule, myosin protein, is the M line right here, and this is these are proteins that are sort of anchoring that myosin molecule in place. So again, now this is a sarcomere from Z disc to Z disc. Right? So now's a good opportunity to sort of pause. That's a lot of information to go back and practice. So let's move on here and look at, continue to try to get a closer look at the sarcomere. So again, here's our sarcolemma. This is one muscle fiber, one muscle cell. We've peeled that sarcolemma away. Here's the peripheral nucleus. And then we've got all these myofibrils jammed into here. And between some of the myofibrils, you're going to have, you're going to have mitochondria. So there's a mitochondrion, and this is jam-packed, a mitochondrion between these myofibrils that are pulled out. So again, when we look at a myofibril, it's repeating units of sarcomere. So this would be 1, 2, 3, 4, 5 sarcomeres, so on and so forth there. So again, here's our sarcomere borders. We go Z disc or Z line to Z disc. And then we're going to see this overlapping pattern here of the actin and the myosin, actin, myosin, etc. So the distance between the Z line and Z line is the sarcomere. We have the myosin molecules ending there, and that creates the I-band. And so the I-band is defined then as between two myosins, and you cross the Z line in the middle. And it was Z line in the middle. The A band then is basically the length of the myosin. So this A band is defined as the length of the myosin molecules. And so this A band here, we have some overlapping of the actin and the myosin here. And in the I band, we just have actin and the Z line protein. So just actin here in the I band. This H zone is where the actin molecules end. And then when you look in this H zone, this is only going to be myosin only. Okay. And then at the center of that, not labeled on this diagram, these dark hash marks here are going to be the M line. Oh, it is labeled, sorry, over here. Here's the M line, which is in the middle of that myosin molecule. Okay? So that is potentially confusing. So this is something that you would want to draw over and over again and practice writing that, practice redrawing that, etc. So again, at the top of this, here's our sarcomere. Again, we're going to go Z to Z. this space between the ends of these actin molecules is going to be the H zone. The length of the myosin, so this length of the myosin here is going to be the A band, and from the end of the myosin here over to the next myosin where it's all just actin, this is going to be the I band. And again, here's your M line right in the middle there. Okay. So I want to look now specifically at the actin myofilament. So we're going to zoom in on this actin myofilament right here. And that actin myofilament, again, is a protein. It's a complex protein, but it's made up of several subunits. One is this rope-like protein that's called tropomyosin. And then you have this other protein that's called troponin. And the troponin is bound to the tropomyosin. Right here you see it bound to it, and here's troponin bound to tropomyosin. And then the purple balls here are the actin molecules themselves. This is another protein, and so the actin or the F-actin molecules here. So again, purple is the actin, the rope is the tropomyosin, and then you have the troponin there. And that troponin is covering these active sites on the actin molecules there. So the yellow here are the active sites, and this is going to be important when we talk about the functionality here in the physiology of this, right? So again, the actin, these are the thin filaments, 3 proteins, right? You have actin molecules, you have tropomyosin, and you have troponin. A little bit more detail about that troponin. There's 3 components there. This is going to bind it to the actin part. This is going to bind calcium ion. Again, this is going to be important in the physiology aspect of this. And then this part here is going to bind to the tropomyosin. Okay. So again, quick look at the myosin myofilament, a little bit more simple. Here it's made up of several units of individual myosin proteins and all bound together. They look kind of like a golf club. So if we come down here, we see that we have, this would be the shaft of the golf club here, and then there are 2 heads here. There are 2 heads to this myosin molecule, and so here's a myosin head, and here's another myosin head here. These myosin heads are arranged such that they are at an angle to one another, and that they will interact with the actin. So the heads of the myosin interact with the actin molecules right there, and right here, and right here in the sarcomere. Okay. So if you understand that, this is a good way to practice this, and we can do this in class as well. So what we have here are cross sections through a sarcomere, and so this cross section was taken at this plane right here. This cross section was taken at this plane. This cross section was taken at this plane, and this cross section was taken right here, sort of at the M line. Okay. And so what I want you to notice, or I don't know if you actually notice anything about these patterns here. And so when we cut through here, we're only cutting right through the actin. Remember that this is again, so I have one sarcomere to there, and then here's another sarcomere to here. So these sarcomeres are lined up end to end to end, and then also jammed into a myofibril, and a bunch of those myofibrils, and then jammed into a muscle cell, muscle fiber itself. Okay. So take a look at that and see if you see any interesting repeating patterns there, and then be thinking about how might this be related to the function, right? So we're going to leave this as an open question for right now. How is this structure of the sarcomeres related to the functioning, that is the contracting nature, the contracting ability of these cells. Okay. Just another image from another text. I maybe click on run these animations. Those are going to help you take a look at it. Micrograph. We have this model in class to take a look at. This would be a model of the muscle cell. Right. So this is one muscle fiber. And again, we have a couple of these models in class encourage you to take a look at them and be able to identify, what are these darker reddish things, what are these thinner ones here, what's the pink, what's the white, what are these red, what's this, what's this yellow, what are these purple guys, okay? What might this be, what are these little yellow things, etc. So have fun with that, and again, thanks for listening. And again, this is Microanatomy 2, where we're looking at the actual muscle fiber here, the cell itself. Okay, bring your questions to class. Thanks. Oh, another one, and a model we could make, and there we go.